In Environmental 200, we did a pollution and seed germination lab that was super fun and interesting to see the different results in everybody's presentation because they were all similar but yet all had their own growth rates and everything like that. So that's what we did this week. The pollutant that we used was a salt concentration mix. We used distilled water that were in squirt bottles in the lab, and then we used salt. I'm not sure the exact type of salt that we used, but we took a certain amount of salt and we put it in these little tubes in the lab, and then you added your distilled water until 20 milliliters, and then you just shook that up, and then you pipetted a certain amount of that salt concentration on your folded up paper towel, which I'll get to. The objective was to mimic the water runoff and salt mixture to relate how it happens naturally in our world, and we were supposed to be checking to see how much salt was too much before it killed the plants. In the reading that we did before we did this experiment, it said too much salt will kill the plants, but a little salt is actually good for the plants, so I thought that was interesting. The design was to take 6 groups of salt with different water concentrations. We did 0%, 1%, 1.5 to 2.5, and 3%. And they all varied with how much salt was in that concentration mix. Then we took 30 radish seeds and we placed 5 wrapped up in a paper towel, and we put that in a baggie, and then we inserted the different concentration mixes into the bags. These were my data tables. They look similar to the other people that were in my group, obviously, because we had the same amount of things. So that's that. You can see that the concentration on the third one, on the 3% concentration mix, never grew the whole entire time, and that just proves that too much salt will kill the plants. But for the most part, 0, 1, and 1.5 took off the most. 1.5 was actually slowing down compared to 1 and 0, and then 2 started to slow down. 2.5 really was pretty slow, and then 3 was none. So that's that. This is just my graph showing I did the measurements in germination growth, and then I did it as every other day, my Monday, Wednesday, Friday. And then in my graph you can see how I did the different color coordinations for every single seed, and you can see that the 3% concentration never grew the whole entire time that it was on there. And the conclusion was that my hypothesis was correct. Too much salt would kill the plants, and a little salt would help them grow, and that just proved it in my graph and in the data tables, because like I said, 0%, 1%, and 1.5% really took off and sprouted really fast, and then after that they all kind of slowed down and to the point where the 3% concentration was too much. And that's it.